Anna is dead, but why? An investigation into the infectious agent inside Anna Garcia by Rue Banerjee, Kaushik Bakuri, and Noah Otto. Growing isolate colonies on agar plates. So first of all, you have to prepare for the aseptic technique. To do this, you wear personal protective equipment, gather your materials, and clean your workstation. Then you have to use the aseptic technique. The purpose of the aseptic technique is to prevent plates from getting contaminated from either the person working on them or the air, and to prevent microorganisms getting onto either the person working on the plates or the workspace. Then you light a Bunsen burner and adjust the flame. You sterilize your inoculating loop by dipping in isopropyl alcohol and then holding it over that Bunsen burner to sterilize it before moving microorganisms from an agar plate to a new one. Then you have to streak for isolation, and you do this by smearing the microorganism on the new plate gently, thinning out the original streak again and again until there are less microorganisms smearing across the plate to increase the likelihood of finding single colonies. And you need to find single colonies because that's what's going to help you with what you need to do at the end, and I'll explain that later. Then you incubate your agar plate for about 24 hours, and when you're done, you re-sterilize your inoculating loop and clean your workspace. So this step is important as a whole because you need to have these isolated colonies because later on you can examine them to further identify the unknown species of bacteria that you're looking for.
So the next step in our process is to perform a gross examination of colony morphology. And we can split this term into three different parts. Gross meaning large, examination meaning observations, and colony morphology meaning regarding physical appearance. So when we put these three parts together, in this step we are basically making large observations regarding the physical appearance of a certain bacteria. And this step is really helpful because it helps us identify possible bacterium that Anna was infected with. So when we are making observations, there are a few traits that we examine, and these include shape, size, margins, elevation, color, and surface appearance. And when we ran Anna's bacteria through the tests, we found that the size was relatively small, it had entire margins, it was raised or convex, and it was colored red. And these traits match up with two infectious agents. The first being Serratia marsicans, and the second being Cocuria rosea. Alright, so the next step is to perform a gram stain. And in this step, we prepare the bacteria for further morphology examination under the microscope. This gives us more information on the structure of bacteria, which I will explain later. So to derive the required results, there are a few steps that we need to take. First, heat the slide of the bacteria colony over a Bunsen burner. Make sure to not overheat it though, as this could affect the results of the test. After heating the slide, add drops of crystal violet to the slide. After this, the colony should be colored purple. Rinse with water and let it stand for a bit. Then, add drops of iodine. After this step, the colony should still be colored purple. Once again, rinse with water and let it stand. Then, add alcohol or ethanol. Here is where we start to differentiate in color. After adding the alcohol, the bacteria colony is still purple, we consider it to be gram-positive. If it's colorless, then we consider it to be gram-negative. In order to further differentiate in color, we add the counterstain safranin. After adding safranin, if the colony is red, then it's gram-negative, and if it stays purple, then it's gram-positive. And the reason this happens is because gram-positive bacteria has a very thick cell wall, and this protects the crystal violet stain from being washed out in the alcohol step. However, in gram-negative bacteria, there's a very thin cell wall, so in the alcohol step, the crystal violet stain is washed out, giving it the colorless color. So we can see how the stain color affects the bacteria and what it tells us about the structure. And in Anna's case, the stain was pink, so the bacteria was gram-negative.
After the gram stain, the next step is to examine the cell morphology. This step helps us identify the bacteria. In this step, the observations we make are whether the bacteria is gram-positive or gram-negative, the shape of the bacteria, and the arrangement of the bacteria. The bacteria can have a shape of either bacillus, rod-shaped, coccus, spherical-shaped, or spirulia, or spirillum, spiral or noodle-shaped. These bacteria can be arranged either in pairs, which is diplo, strepto, long chains, staphylo, clusters, tetra, groups of 4, or sarcina, cubes. The nomenclature for naming a bacteria once it's observed is its arrangement plus its shape. For example, if I have a bacteria that has a diplo shape, and if it is a bacillus, it would be called diplobacillus. The bacteria found inside of Anna was a gram-negative Staphylococcus.
So biochemical tests. So what this step does is that you're trying to see how the bacteria eats, breathes, and produces waste. And that's necessary because it can be used to determine the exact species of bacteria, and we need to know the exact species of bacteria to help people's health and help help them fight off these diseases properly in the most efficient way possible. And when I went through the charts and I compare all the information we have, uh, the type of bacteria that Anna was infected with is called Serratia marcescens. Serratia marcescens. So overall, biochemical tests are just a very important step because the charts can lay it out in a very simple, straightforward way that can easily tell doctors how to narrow down what type of bacteria the person they're trying to treat might have, and if they know that, it can help them greatly in trying to treat and help the person.
Based on our tests, we can conclude that Anna was infected by Serratia marcescens. She had a urinary tract infection, which was most likely received from a contaminated catheter. The next steps are to administer adequate antibiotics such as aminoglycoside and anti-pseudomonal beta-lactam. The reason that Serratia marcescens is dangerous is because it can lead to bacteremia, which is an excess amount of bacteria in the bloodstream, and septic shock, which is a drop in blood pressure, which can lead to organ failure. Septic shock is caused by sepsis, which is a widespread infection in the bloodstream. The reason that this could have killed her is because the signs of Anna, a lack of oxygen, and that she probably had collapsed, could have actually been caused by septic shock.